Colloquium this week. Very kind of you. I don't think that I know all of you. My name is Everett Schock. I'm a member of the faculty in the School of Earth and Space Exploration. What a surprise. Today we have a special opportunity to think about life a little bit, or aspects of geobiology, which for some of us is a very exciting thing to contemplate, because many of the environments and processes, geologic environments, geologic processes, planetary processes that are at a lowish temperature tend to be populated. And the argument goes that perhaps first of all, we should not ignore these parts of rocks and soils and atmospheres and around us, water and so forth. But also, it's not quite clear still in the sort of early years of geobiology to what extent organisms, microorganisms especially, in various environments are hanging out, doing what they can with what's being provided to them, just sort of like passively and hanging out in the systems in which they're found, or to what extent are they actually driving what's going on in those systems. So you might be obsessed with something like surface processes, soil development, geomorphology, things like that. All of those kinds of conditions you're fascinated with are probably populated. Ooh. To what extent are the organisms in those systems just along for the ride or driving the bus? Things we don't know. Or you might be obsessed, like some people are, with hydrothermal systems. Very strange people. Thinking about, we got all these hot areas of the earth that are cooled by circulating water. And what's up with that? Those systems in the lower temperature reaches tend to be populated with all kinds of life. Are those things just there, passively, if we could ignore them, like historically we've done in the geological sciences, and that's okay? Or are they more directly involved? Or you might even be concerned about the atmosphere or interested in things going on, and that's what we're going to hear about today. So that was a segue. All right, so today we're going to get to hear about what's going on with the atmosphere. Maybe there'll be some geobiology, and we get to hear all of this from Professor Elizabeth Trembath-Reichert, which is it's a great pleasure to introduce. Let's see, undergrad, Barnhart College, graduate student at California Institute of Technology. Oh, in between worked at NOAA, and a postdoc at the Woods Hole Oceanographic Institute before joining the faculty here in the School of Earth and Space Exploration. Take it away, Elizabeth.
Thank you. All right, so the title of this talk is a little tongue-in-cheek, because there's sort of this long-running joke amongst the junior faculty that about every month or so, someone will mention that no one in SESE studies the atmosphere. And so I just wanted to boldly declare that my lab group studies the atmosphere, and you're actually going to get 3 atmosphere talks in vaguely maybe ascending order, with the one we had on ozone last week, and then we'll have the postdocs next week, and then Katrina will take us all the way up to the outer limits of the atmosphere with her talk as well. Okay, so a little brief outline. I'm going to give an introduction to aerobiology specifically, why we should care about it, what are some big ideas that we need to address. And then I'm going to do something a little maybe unique. I'm going to talk about the journey to actually get to even work in this field and what that looked like. And then I'll get to the areas of aerobiology that we've been exploring at ASU in a bit more detail. So, as Everett hinted to, understanding global biogeochemical cycling might be a good reason to look at the atmosphere. And we're pretty okay with the idea that atmosphere probably has some carbon in it that comes up a fair amount. And there's more carbon stored in the atmosphere than in plants. And we're pretty open to the idea that CO2 might be an important aspect of this. And we may even be aware that that's how plants build their biomass, is by taking in CO2 from the atmosphere. But that's actually only one very small part of the vast metabolism of microorganisms that can live off the air. And so here's actually a paper that just came out in May of this year. And so what they did is they filtered down a bunch of organisms that essentially grow on leaves. And then they fed them different trace gases. So they fed them methane, they fed them carbon monoxide, and they fed them hydrogen, just at levels that are in ambient air that we're breathing right now. And so from time 0 to 6 months later, this is evidence that colonies grew up on the filters that they filtered them down onto. And then this is also evidence that they were able to draw down the concentrations of methane, carbon monoxide, and hydrogen over about a 1-month period. So organisms that are just sitting on a filter can do this process. And so microbes can metabolize a whole range of gases. It's not just about CO2. But what we don't know is if they can do this in situ. So is this actually happening in the atmosphere? These are all laboratory studies. And then some of you may have seen me give this slide before. I'm going to give it for a different purpose now. So this is an estimate of the number of cells in the atmosphere, which is 10 to the 24th. And that's on the same scale as the number of stars in the universe. And so even tiny rates, so even if there's only like 5 organisms doing this per cubic meter, there's enough of them up there that this could be significant on biogeochemical cycles on a global scale. And then there's sort of the more maybe astrobiology side of this. There's a lot of extremes in the atmosphere. So there's a general idea that microbial occurrence will decrease as we go up towards the stratosphere due to a combination of different stressors, right? So you have radiation stress, you have temperature stress decreasing up to a point, you have decreased pressure as you go up, and you're going to have less water availability, and you're going to have less nutrient availability, and then you're going to have this ozone issue that was talked about last week. And there might be some sort of pockets of protection, like maybe if you're in a cloud, you might have it a little bit better off. And so the overall assumption is that the likelihood for survival will decrease with increasing altitude, but the exact limits to this are not known. And what we're really after here is trying to declare the atmosphere as its own ecosystem. Because if we can convince people that it should be, it actually would become the Earth's largest ecosystem, outpacing the ocean by about an order of magnitude. So it's very important that we characterize what life is there, how active it is, and what roles it plays in global ecosystems, geochemical cycling, et cetera. And we're not going at this completely blind. We know that cells have many adaptations to the various stressors I just listed. So we know that cells can form spores and go into essentially like a hibernation state. We know that they have ways like making this nice EPS goop, if any of you have worked with biocrusts in the desert, that provide like a sort of collection for moisture and a sunscreen. We know they have all sorts of crazy ways of changing the osmotic conditions within their cells to protect their proteins and keep everything functioning. So we know they have the capacity to survive in the atmosphere. Here's one example. So, if we're trying to think about, Everett mentioned that these surface processes are likely what would be seeding the atmosphere, then there's probably some parts of environments that exist on Earth that are going to pre-adapt a microbe to be able to withstand those stressors that I just indicated, right? So one example might be Antarctica. It's pretty cold, it's pretty dry in many places. So here's a study looking at colony forming units, or CFUs. This is just like amount of microbes that are not dead. And here is a mesophile, so it likes conditions that we like. And then here's this Antarctic isolate. And so when they just took the culture on the ground, everything was fine and happy. When they then just shipped it to another place, everything was fine and happy. Maybe slightly less happy for the Antarctic one, but within standard deviation, it's fine. And then when they put it up on a stratospheric balloon, suddenly the mesophilic strain tanked by orders of magnitude, whereas the Antarctic strain had a much less, much smaller reduction in survivability. And so this is hinting this idea that there are organisms already on the surface of the Earth that we know are pre-adapted to dealing with atmospheric conditions. And how cells enter the atmosphere actually might play a role in survival. So for those of you that think about surface processes, this is a pretty interesting study that compared aerosol formation by bubble bursting. So a microbe is here, it gets caught up here, things pop, and then all of a sudden it's instantly dried and aerosolized, as opposed to taking a droplet and placing it on a slide and then just letting it slowly dry over time. And so this is, again, a measure of how many cells remained alive. And you actually can't even see how many cells remained alive from the surface drying when you try and plot it with the ones that were surviving when it's aerosolized. So how fast you dry it is actually a really key feature of survivability. So there's, physical processes that are going to define this process, and then there's also going to be, metabolic adaptability as features that we can point to in trying to figure out how the aerobiome might work. And then this is actually one of my favorite studies that I've come across, and I'm going to try and make the case that there are vacuophiles. So this is a Siberian permafrost isolate that actually grew better under low pressure, no oxygen conditions. And so this was, devised as a study to look at Mars atmosphere. So that's why they picked 7 millibars, but that's equivalent to being in the Earth's stratosphere. And so here's the evidence for this. So here in A, they plated everything out with oxygen. And then here, everything just has a CO2 atmosphere. And then here, everything has a CO2 atmosphere, but down to 7 millibars. And so, there's some colonies that preferred growing under those conditions. It's barely present here. It's significantly present here. It's even more present here. And then here you can see it in CFUs again as well. So way outperforming by like an order of magnitude when it's given this lower pressure CO2 atmosphere. So there may actually be evidence for organisms that prefer to grow in the atmosphere rather than just withstanding the conditions available there. And I also want to bring up that this is actually not a very novel endeavor. So for some historical interest, you may not be aware, but Amelia Earhart was actually one of the first aerobiologists. And so this is a paper from 1937 lamenting the loss of the aerobiology collection that went down with her plane. And so she had a series of instruments where out on where the pilot could open and close these tubes, there was a glass slide covered with petroleum, sterile petroleum, like a Vaseline essentially. And then when they got to the point of interest, they could open the tube, expose it to the atmosphere, and then sterilely close it back and return the samples when the plane returned, was the goal. And from some earlier expeditions using this method, they were able to show that, for example, when you flew over the Arctic Ocean, you were able to find diatoms and marine organisms in the atmosphere, as opposed to when you flew over land, you found pollen and things like that. So even going back to the 1930s and 40s, there's a pretty good idea that there were microbes in the atmosphere and there was a correlation to the surface processes that would be seeding it. Okay, so hopefully you're now all jazzed that aerobiology's got some cool stuff that we need to figure out. And so then I'll take you on the journey to get to be able to study it. So I started ASU in 2019, and my PhD lab was known for studying methane cycling in marine sediments and carbonates, so ocean, stable isotope probing, and greenhouse gases sort of were the main ideas there. And then I went on to do a postdoc in a lab that was known for sequencing organisms in hydrothermal vents on the seafloor and then culturing them. So again, oceans, stable isotope probing, and a little bit more of a genomic focus. However, in both places, I actually studied what life existed beneath the oceans. And so I was focused primarily on the deep subsurface with stable isotope probing and genomic components. And so the question I was faced with is, how do I go from this to this in areas that I would like to look into? So there was a motivation for this. So right around the time that I was finishing my PhD, most of the funding mechanisms for deep subsurface work ended. And then there's sort of a general idea that when you start your own research program, you're supposed to kind of have some unique themes that make you look a little bit different than what your PhD and postdoc advisors are up to. And so both of these things kind of, shoved me in this direction. And so the question was, can I use my methods that work in the deep subsurface to study life in the atmosphere? And the only problem is there's no dedicated funding program for aerobiology. And so this necessitated a two-phase approach. So phase one, I tried to fund work in dryland soils where organisms have similar stressors to the atmosphere. And then in phase two, directly seeking funding for that aerobiology work. So what this ended up looking like, starting with the Department of Energy. So they have this environmental system science program, which examines complex ecological, hydro-biogeochemical processes within terrestrial and coastal ecosystems to understand emergent properties and changes to Earth and environmental systems. It does not mention the atmosphere, and they mean that. So no interest in the atmosphere confirmed. I actually don't have a slide on this, but I tried again in 2024, and I somehow managed to convince poor Alexi to apply to a program call that the program officer called me up and said, how did you possibly find this? This is actually a program call that doesn't even exist. And so we definitely saturated the DOE options there. Okay, so then Maitri and Heather got to go on this journey. So we tried one of these eager proposals. And so these are actually supposed to be targeting high risk, high research payoff, kind of novel new things. So we submitted a one-pager to program officers there in 2020, and then also submitted a full proposal to the geobiology, low temperature, and geochemistry. And so they specifically support research on Earth surface processes. They're okay with it being land and not so much the ocean because that's somebody else. But again, they didn't specify anything to do with the atmosphere, and they meant it. And so in one case, it sort of was considered too risky and undefined, and then proposing too much in order to overcome how risky it was. So those options didn't really pan out. So let's go on to exobiology. So here, the program is to understand the origin, evolution, distribution, and future of life in the universe. Atmosphere, I believe, should be included in the universe. And so research centered on anything in the universe seemed to be fair game. And so we submitted a full proposal in 2020 there, and it worked. So NASA gets it, and phase one is complete. And so to tell you a little bit more about that project, so this was a project called Cold and Dry Limit to Life, Understanding Microbial Activity in Dry Permafrost Samples from the Newly Discovered Elephant Head, Antarctica. And so this is a really fun project where I get to work with Dr. Mary Beth Wilhelm at NASA Ames and Jackie Gordial, who's a professor at University of Guelph. And they both have way cooler pictures than me when I Google image search them to find stuff for this talk. So you should Google them. They're pretty awesome and have a lot of really great outreach information. But the main motivation for this project is that these dry permafrost areas in Antarctica provide some sort of analog somewhere between past Mars and present Mars, and have these dry permafrost conditions, but are not known to exist anywhere else on Earth. So we got some samples from this. It's called elephant head because this vaguely looks like an elephant's head. There's the eye, tusk coming out there. So we got some dry permafrost samples coming back. Actually, during the pandemic, they were able to collect them and send them to us. And so this paper actually just got published this year. And we had a really interesting story where it looks like, so from Jackie's work during her PhD, she worked in the University Valleys. So that's most of where the sort of NASA focus had been on dry permafrost. And so if we look at a cumulative time and temperature plot versus water activity of ice, University Valley spends cumulatively more days at a colder temperature than Elephant Head. And so here's a picture from Jackie's sort of major thesis work where she grew dry permafrost samples and at -5 degrees Celsius, so the upper 5 centimeters of core. And she basically, and this is a proxy for activity, she couldn't see any difference from the killed control versus her live sample on this activity proxy when grown at -5 degrees for 600 days. However, from Elephant Head, we actually were able to see a difference from killed control. So these are our two live samples, and then the round circles are their relative kill control. You can see the activity is even lower. So we're at 1 here, whereas 1 is barely showing up here. But we were actually able to show that dry permafrost is not dead. So prior to this, dry permafrost was considered completely dead. This work showed that it's not. So dry permafrost is not dead, and it looks like there's really this razor's edge that might just be this like 5-degree difference between how many days you're spending cumulatively at -10 versus -5 that is allowing for that distinction. So really fine-tuning that sort of razor's edge for cold limit to life. And I'll just say that I'm going to stop talking about this because Chanel will soon tell you more about what our lab has been doing in this space when she, gives her magical defense talk in a year or so. Okay, so got to go back to phase 2 here. So there's various other programs that go for, just propose us something. And so this is the Beckman Young Investigator Award. And so this is just vaguely chemical and life sciences. So proposed to that one, made it to the final round in 2021. And then it turned out that they just kind of thought it was interesting, but really wanted to fund more medical stuff. So that was a no. And then some other folks at ASU, we proposed to the Keck Foundation, and which also supports these sort of like new novel, projects that, have no other way of, and they're going to change paradigms and all that kind of stuff. So for that one, we made it to the ASU selection level also in 2021. So, some more X's, but maybe getting warmer for looking for these types of funding sources. So then a team of early career researchers, we went after the Early Career Award for the Human Frontier Science Program. And so that promotes international collaboration in basic research, focus on elucidating sophisticated and complex mechanisms of living organisms. So they don't care where the living organisms are, they just need to be there. So we made it. So in 2022, we finally got funding. And I think the takeaway was we needed to make it more clear we didn't know what we were doing, because this funding program, actually, you have to work with people you've never worked with before. You have to work on an idea you've never worked on before, and everyone has to be from a different country. And so, we were just what they were looking for. And so it's actually a really cool funding program. They also have a postdoc program so for any of you that want to do an international postdoc, I highly recommend looking into these awards. It's a really cool organization. And I'm not just saying that because they funded me, but okay. So work finally begins in 2023, and then the first talks on this topic are finally happening in 2024. So some kind of takeaways from this: new research directions are not quick to easy to get going, and you got to embrace the side hustles and find ways to infuse other aspects of your work with what you want to explore. And finding your team is really important. I have lots of great colleagues here who are willing to take some risks and try and try some of these proposals with me, and engage proposal writers and readers make a huge difference in, keeping the motivation to keep this going. And some funding organizations and reviewers are more amenable than others, but they can escalate change based on community action. And so, I maligned NSF maybe a little bit in this depiction, but actually they're putting together a workshop in February about the need to fund aerobiology. And so enough different groups have tried to sort of get these types of things funding that they're like, oh, we should fund this. And so that, community action can change where funding organizations want to spend their time and money. Okay, so now I'm going to tell you a little bit more about the aerobiology work we've actually been doing at ASU. So here's the project. You will see Jackie is here again, which suggests they're a little open on the having worked with them before side of things. So she's the lead PI, and our overarching question is the atmosphere a living, breathing ecosystem? And then we have 3 sub-questions. So first, our atmosphere community structured and adapted, led by Chris Greening's lab at Monash University. Then we're taking on the metabolically active question. So that is it happening in situ question, that's us. And then we have James Bradley's group looking at the modeling the amount of energy that might be available for life in the atmosphere, and hopefully extending that onto exoplanet work. So if any of you are interested in that side of things, I'd love to talk more. And there's a really motivated postdoc who's taking that off to new heights now. Okay, so this project had a very unique amount of calibration required. So this is a figure from a review paper that we put out this year. And aerobiology has a lot of contamination control issues, a lot of low biomass issues, and the general sort of lack of standardized sampling. And so if we're going to make some claim that we're going to be able to do a global survey, then we all need to A, divide up, and B, agree that we're all going to do it the exact same way. And that's not something scientists necessarily are always very good at. And so we actually had a meeting last summer up in Flagstaff where we all got together, agreed on our protocols, figured out our contamination control. So you can see we're all wearing clean room suits out in the environment because we are the problem. Like, we are contaminating our samples. And so we all agreed upon how to do that. I think this might be some postdocs that are in this room. And then, so in order to get the global survey underway, we agreed not just how we're going to do the contamination control side of things, but we're going to take soil samples in cardinal directions. We're going to measure all these different metadata, et cetera, et cetera. So this team got together. We're ready to head out to our respective environments. Here we have two of the sites that we've focused on in Bermuda and at the Santa Rita Experimental Range, where Heather also does a lot of great work. And then we have our folks that are more focused on polar regions, who tend to beat us for the most epic photographs, but are collecting samples with a bit of harsh conditions. So here's a table of ecoregions, and we've covered almost all ecoregions by at least one sample. So the black ones are the ones we're still missing. So if any of you have field programs in these places, let us know. And we've covered about half of them with at least 3 different sample types that fall into that ecoregion. So we're trying to not just go everywhere once, but to actually have repeated samples from, North American version as compared to an Australian version. Okay, so then I'll talk a little bit about the work that Alexei Besser's been doing as a postdoc at ASU. And so here's some of her data looking at the different ecoregions. And so from these filters that we go out and collect, we can see that some ecoregions have they have different concentrations of airborne DNA because we sample them all for the same period of time, and they follow general accepted trends. So it's, for example, known that marine samples have much lower biomass in the atmosphere above them as opposed to terrestrial samples, and so that checked out, which is great. But then there's also some variability depending on time of day, right? So here's some work where she got to have fun getting up at 2 o'clock in the morning, sampling till 6:30 in the morning, then doing it again at 7, going to 11, then doing it. Yeah, so definitely put in the work, but was able to demonstrate that if we just went out and did everything at one time of day, we would miss a lot of this diurnal variability as well. And so it looks like this sort of morning period when everything is getting up and becoming active is also when the atmosphere is more active as well, or at least has a higher concentration of microbes. And then this is just for the biologists, but she spent a lot of time trying to get PCR to work. And so we have great results that on this gel here, basically if you see a bright bar, that means we can actually amplify our DNA. And so if you're dealing with low biomass samples, can you even get enough DNA out to sequence is an open question. And this was confirmation that we were able to get that working. So good hopes for being able to actually survey who's there, not just if anything is there. And some of our samples come from the roof of this very building. Stay tuned. Okay, so the single cell side of things. So this is a shout out to BioRender for those of you who use this program. This is a pretty fun figure to put together. So the basic idea is there's some general conditions that are going to affect everything in the atmosphere no matter where it is, right? Like we know temperature is generally going to decrease. We know pressure is generally going to decrease, at least the parts of the atmosphere that we're going to be able to sample. But there might be some variability that's on the micro scale, aka the microbial scale, right? So if a cell is in a rain cloud, it might be surrounded by water particles, right? Or if a cell is in a dust cloud, it could be surrounded by dust particles, which can actually attract water. And both of these cases are going to provide radiation protection. So if you're in one of these microenvironments, you could have reduced radiation, increased moisture, and increased nutrients at the cellular level. And so Q, single cell processes, being important. And so this is work that John Lemus was doing as a postdoc here at ASU. Now he's gone on to work for the Johnson Space Center, but still doing nanoswim's work, which is very exciting for this project. And so he put together this high elevation research and astrobiology platform. And the reason why we actually had to put together a new platform, which also confounded some of our attempts to get funding, was we needed these special conditions, right? So I just told you this whole other project where we went out and just like filtered air, like that's doable. But we needed to be able to filter air in a way that we could look at it for microscopy. So we needed everything to be on a flat surface, and we needed it such that all the particles weren't jumbled all up on top of each other. And so most of the things that existed were made to get you as much sample as possible, as fast as possible. And then we also needed to be able to deliver our stable isotope of interest. So he put together this system that has an electric three-way ball valve, some compression fittings, and then two custom filter cassettes where we can lay our 0.2 polycarbonate membrane filter on it for our sample collection. And the basic idea is that we have a sample collection phase where it's open to the outside environment, pulls air in and sends it to these two filters. And then we have an incubation phase where we close it to the outside environment and then can pull in isotope labeled gases that can sit on the filters to expose cells on the filters to the label. And then we can vent, return to the initial condition, vent the label, and then return the sample from our study site in situ back down to the lab. Okay, so I've hinted to this idea of stable isotope probing as being important for these types of activity measurements. So I'll go through that now. So the basic idea is the main constituents of a cell can be broken down essentially into this chemical formula here: carbon, oxygen, hydrogen, nitrogen, phosphorus. So if we label any of these with a unnatural amount of any of the isotopes, then we can use that as a tracer as to how cellular life is building its biomass. So let's take deuterium, for example. So natural abundance deuterium, pretty low concentration, 0.01%. So for a given incubation condition, a cell is going to be a tiny, tiny bit of deuterium at natural abundance. However, say we create some experimental conditions where we're growing them in, say, 10% deuterium, then anything that is being, any biomass that's being synthesized during that incubation is going to take up that deuterium label. So the cell will become labeled. And then you can do different things with it, right? So you could filter all those cells down, look at the concentration, the total isotope concentration of all the cells. You could pull out specific biomolecules of interest, right? You could look at what portion of the DNA is labeled, what portion of the lipids are labeled, what portion of the proteins are labeled, because then you get that additional taxonomic and functional information. Or you could look at the isotope concentration of single cells, which is what the NanoSim instrument here at ASU allows us to do. And so here's an example of that. So here is the hydrogen ion image. So this is like 5 different cells that we're able to detect here. And then in this case, 2 of them also took up that deuterium label. So 2 out of the 5 are active, and we can count individual single cells and be able to determine this information with this technique. So we got labeled into some cells that came from a filter. So we have carbon-bearing enriched particles detected with nanosims. So this is a filter that was actually just filtered in lab air. And so here's a measure of the deuterium incorporation from nanosims, and then also going back with SEM and showing that they are carbon-bearing particles as well. And so this is kind of like the project's at the stage of like, we can get some cells on a filter, we can get them on the nanosims, we can see the deuterium isotopes are labeled, and so we're going forward from there. And the eventual plan is to have this pipeline where we take one filter and take it down and do particle identity assessment with SEM, and then take it onto the nanosims, and then we can also do other sort of DNA staining and fluorescence imaging. And the main issue here is every type of stable isotope probing that's basically kind of ever been done before has some ability to wash afterwards. But I'm building this whole system around the idea that we're trying to keep things like the atmosphere, and the atmosphere is not inundated with water. And so we're trying to maintain a system where we never have to add water throughout the whole process, but we still need to be able to wash and remove this label. So this is kind of like the part that we're stuck on right here. So if any of you think about how deuterium moves in different particles and want to talk about this more, please let me know. Okay, so then I'm going to talk a little bit about the sound side of things. So visual observation is not as helpful for the atmosphere as it might be, say, for like a hydrothermal vent on the seafloor. You know, you look up right now, and you probably can't tell me what microbes are there, but sound is actually a more appropriate medium for telling us what might be in a room, right? Like I can hear voices, I can get a sense of how many people are here and what they might be doing. And so we actually have been including a sound recording, field recording element of this. So here's some different sound maps that Alexi and I made when we were out in Bermuda, thinking about all the different sound sources we were hearing, while also making parallel recordings to draw attention to the different sources of bioaerosols, like leaves, birds, bees, etc. And so I actually have some sound recordings for you. So I'd like you to think about, as I play these three, which source would you generate more microbes? Which source has a higher chance of long-term activity in the atmosphere? And then what human-induced sounds do you hear? So there'll be three clips about a minute long. One. All right, and then the last. Okay. So here's what these sites look like. And so to go through these ideas, so which source would generate more microbes? Who thinks it's A? Yep. Got some votes for A. How about B? Okay, some votes for B. How about C? Oh, people like C. Okay. I would probably say B or C. And so A is actually getting most of its air from the ocean. And so this is marine sourced and has a lower concentration. Whereas B and C, it kind of depends on the time of day and maybe other conditions as well. Which source do you think has a higher chance of long-term survival in the atmosphere? A. Okay, a couple for A. How about B? B. How about C? Okay, so that's pretty evenly distributed. We don't, and actually, I think that's a fair answer because this has lower density and it might be used to salt, right? Or some sort of water activity stress, even if it's not water stress. Here, it's going to be dry lands, right? So they're going to have radiation, maybe temperature extreme stress. Whereas here, it just might have a ton of stuff. And so therefore, just by like winnowing, it might actually maintain a lot of microbes in the atmosphere. So that's all fair game and about where our research is. So probably could be B, but open question. Okay, so what human-induced sounds did you hear? Walking? A train? That damn humming? All right. Yeah, so sampling towers are really loud. And audio draws your attention to that, right? So if you're just looking at this, you're not like, oh my God, we're horribly disturbing the environment. But if you hear what these places sound like, it totally changes your perspective. And so this is actually forced us to think a little bit more about how these different sampling towers are affecting the ecology that we're trying to measure. So this is actually part of this larger project that we're getting to work with, this Language of Landscapes project. And so this is a project that has community engagement activities where participants make field recordings and then work with musicians to assemble compositions representing localized sonic ecologies. And then the musical composition is produced by composers. And then there's this cello percussion duo, New Morse Code, with Michael Capitello, who's a professor at ASU, and Hannah Collins. And so these groups are actually already going out and doing this community engagement work. And then the hope is that we can sort of feed into both the continued community engagement activities and then also this public digital resource to encourage climate change action through engaged collective observation and communication. And so I mainly bring that to the forefront because stay tuned for a collaborative event with our HFSP team, Language of Landscapes, and the Phoenix Downtown Chamber Series this spring. So with that, I'll take your questions. Thanks.
How did you stay motivated to keep like wanting to get funding over that long period of time after like I assume I've never put a proposal together like that, but the rejections probably hit pretty hard because they're probably not like the reports I do like the night before they're due. So
well, I don't know. Everett told me he can't go to dinner tonight because he's working on a proposal that's due tomorrow morning. So so I think that's where the team comes in, right? So when I mean, this is like good for when you're writing a paper, your first paper or something like that, like when you just never ever want to look at it ever again, if you have someone else who will talk with you about it and give you that new perspective and make you think about it in some way you hadn't been considering, then it gets you excited about it all over again. And then I can be a little cheesy about all the folks that work with me that are sitting out here. Also, having students that are really excited about these ideas really helps, right? Because it's like, I want to enable them to get to do cool stuff. So, your peers and your students help keep things going. Yeah.
That was great. Thanks. So one of the surprising things for me, I've always thought of the atmosphere as being extremely well mixed, and both what you were talking about with your work as well as going back to Amelia Earhart's work.
A sentence I never thought someone was going to say about what I do, but it's great.
But it seems like you've got very distinct microbiology over different source areas rather than it being really well mixed. And why is that? That seems counterintuitive to me.
Yeah, so I think there's also, so I guess most of the research on atmospheric dynamics and particles comes from atmospheric chemistry interests. And so a lot of that is based on like particle size, right? So if we look at what's known about particle size models, 10 microns looks like it's a pretty important distributor or determining factor. So anything that's like 10 microns to 100 microns is actually probably going to fall back out of the atmosphere within like days to a week. And so you have to be really tiny in order to make it for a while, make it across the, the Atlantic Ocean. And so, and then there's likely some correlation to survivability in there, right? So like if you're just a lone microbe trying to jet across, that's probably not going to work very well for you. But if you're in some particle that's like protected and attached to, other microbes and sources of nutrients, et cetera, maybe you have a better survivability rate. So there's kind of this interesting trade-off between, size and transport capability and survivability that I think is an important dynamic there. And then there would also be the idea of, and there's all the stressors that we just talked about, but there would likely be some sort of differences in the dynamics of how, just even, I just showed you like time of day can make a difference, right? So even in the exact same place, sampling for 4 hours, every 4 hours apart, you're going to get totally different answers. So when and where make a huge difference. Yeah. Oh, it's a race. All right, you got it first.
Okay. So when you're taking these sorts of samples out in the field, you are taking these steps to reduce human impact on those samples. What is the general distance you want to be from other humans? Like you're up here in this, I assume this is up in Flagstaff.
This would not be how you want to sample, I guess.
Yeah, but how far? Because you can only get so far away before you start running into humans again. So what's the general distance you try to aim for?
Well, so this is sort of a philosophical question almost, where it's like, what is a pure sample, right? So if we are interested in describing how the atmosphere works, then we're somewhat agnostic to where those organisms are coming from. We just don't want to be the source of the organisms, right? So if it's there because there is a fertilizer plant downstream that is going to be a constant source that is part of that ecosystem now, then that's sort of one thing. But if we're just sampling because we walked in, kicked up a bunch of dirt and breathed on everything, then that's, we change sort of that longer term kind of scale dynamic. But most of the places we've been focusing on have been remote, but we do have urban paired samples for all of our study teams. For example, we have samples from the roof of this building. So yeah, we're not trying to remove all human impact. We're just trying not to be the human impact.
Okay, cool. Thank you.
Yeah.
So I was aware that we had people studying the atmosphere, but I wasn't aware that one of them is also doing seismology.
Yes.
So I wasn't sure though, are you showing that, because it's like acoustic energy, or you can think of it as like a mechanical energy. Is that like a form of stressor? You could have thermal stress, chemical stress. So is that something that you're studying? And is there, because there's a lot of seismology you might be able to do in those recordings, like looking at spectra averaged over time, and some frequency range corresponds to events that might be stressors. Was that a motivation, or were you just curious, like, hey, let's put a microphone out there?
Kind of the latter, but so I mean, this was informed by colleagues at ASU who do acoustic ecology. So that is their body of work. And so, but as far as our personal involvement in it, it was a little bit let's see what we find, because sound seems like a better medium to understand our system than sight. So, so that's kind of where it started. But then it wasn't until we listened that we realized how big of a deal the human impact side was. And so the thought there is, I mean, there are examples where it is known that animals won't be someplace because of vibrations, right? And so if we're trying to, again, think from an ecological perspective, if there's something that's preventing animals from wanting to be there, that'll change vegetation patterns, and that would therefore also change the microbiome of that ecosystem. And Alexi actually has some data already showing that the soils around these sites have a different composition than soils that are like, you know, what, 50 meters away, 100 meters away, kilometers away. Yeah, so in the same, you know, climate, being next to the field station versus not has a different soil microbiome. And so we are more attuned to trying to think about art, but the atmosphere might be less affected, right? Because it is some amalgamation of across a kilometer scale. So we're just trying to figure kind of like where we fit in that space of ecological processes and atmospheric transfer processes to try and pinpoint what these differences might be. But yes, if you have ways to pull out sound information that could be interesting, we'd love to talk more.
I was just
Oh.
curious, sorry, way back
You're in the
there,
dark.
if you don't mind. The studies that you mentioned early in the 30s from the flights, like for example, Amelia Earhart, has anyone gone and then taken those analogous samples to see how things have changed in the 100 years, approximately 100 years?
I don't know if people have done historical studies because, well, I guess my guess is well, I can go and say, I would imagine the preservation of those samples is minimal from a microbial perspective. I bet the diatoms could still be there because they have like a silica crust. So like, I don't, you know, they weren't put in a -80 freezer after the flight landed. So I imagine their sort of examination was a pretty much kind of like one and done type of information. But as far as like thinking about the general patterns of what you are seeing in samples from different places, I don't know, maybe you saw there was a news story that came out this week of this research in Japan looking at the fact that potential pathogens can transit across the country in the atmosphere. And agricultural interests have been really interested in this for a really long time, right? Because they know that one field can be fine, another field can be completely inundated with some sort of airborne, you know, fungus or something like that. And so they really want to know what sort of transit. So there's a lot of human health and agricultural interests that have sort of motivated that early aerobiology work that continue to today. So that's probably the literature where I would imagine that type of comparison is. Yeah, I guess what I'm trying to say is I think those comparisons are going to be very targeted towards potential human issues or potential agricultural issues rather than this sort of more ecological perspective. So yeah, I think it would be worth revisiting that from this larger ecology as opposed to maybe those more sort of targeted interests.
So I'm really intrigued by the last thing you talked about, the language of landscapes and the objective of this. Are you trying to characterize different landscapes by a sonic mix or a sonic signature or something? Is that the goal of this project?
So, well, okay, it's not my project.
Right, now you're involved, but yeah.
But Mike is sitting right back there, so you can bug him about it later. But so I think from discussions I've had with him about this, one of the main things that they're excited about is the way in which music can draw attention and draw focus to an issue and get a community to come and be in the same place and experience a similar moment together, right? Like that's what music does. It brings us into a room, we're all gonna listen, we bring our separate experiences, but we're all currently listening to the same thing. And so they took that to climate scientists to try and draw concentration on climate action. And so we're interested in both using scientists as that community and helping us think about how we do science. I mean, I think this idea of listening to the towers is a great example of like, we've already sort of thought about things in a different way than we already have before. So some of it is sort of like adding us to the community and kind of having this co-evolution of artists and scientists thinking about a project from its sort of inception. So we're pretty excited about those ideas. And then, I don't know, do you want to add anything? Great, okay. Did that answer your question?
Yeah, thanks.
Okay, okay, yeah. Yeah.
Okay, hi. Over
Yeah.
here. So my question is, do you think microbes, I realize this is maybe a bit more like conceptual question, but do you think the small molecule chemistry that microbes can do affects the chemistry that are in clouds, which long-term affected what could be occurring on terrestrial processes or just on the Earth?
Yes. So I mean, this comes up a lot, right? Like I'll give a talk like this, and then someone who studies the atmosphere from a totally different perspective will come up and be like, I've never thought about there being something alive there, right? And so it's really exciting to think about the ways in which just simply merging data sets that already exist could be like an immediate win. And so actually the modeling team that I mentioned on the project is doing that. They're like simply synthesizing the types of data that already are out there to try and understand what might be sort of unexplained or missing patterns that microbes could fill in the gaps. But I think right now it's kind of a black box, right? Because the ways in which we're measuring things about the atmosphere, there's this idea of cryptic cycles with microbiology, where if you're just measuring the two end products, but microbes have turned that over 50 million times in between, you would have no idea, right? Because the net state is no different. And so there either could be possible cryptic cycles that are not like moving the needle as far as budgets that are really important, because if it were to stop, then you might finally see something, or there could be more critical thinking about sort of the different parts of the box model that we're measuring in different places, and sort of where would be the most likely places to elucidate a potential microbial role. So it's something that we're very interested in looking into, and I think it is highly likely that they're having an effect that we're not appreciating. Okay, well, we're at 4:30, so I will free you all. I'm happy to take additional questions. Thanks.